a little bit different representation. Prophase 1, remember in prophase of mitosis, what happens? The nuclear envelope starts to fragment. Well, that happens in prometaphase. But the chromosomes all condense, and the mitotic spindle starts to form. Now I have a meiotic spindle. And you can see that these pairs of homologous chromosomes are starting to line up. So in mitosis, if this was mitosis in this cell that had 6 chromosomes in 3 homologous pairs, there would be 6 things lined up here at metaphase, and not these 3 sets of 4. There would be 6 sets of 2, not 3 sets of 4. Okay, I'm going to talk about these chiasmata and what's happening there later on. But for now, what's happening is at metaphase 1, the homologous pairs are lining up at that imaginary metaphase plate. In anaphase 1, the homologous chromosomes are going to separate. And at telophase 1, we've got 2 haploid cells. And you can see they've all got a mix of blue and red. And that is significant, but I'll tell you more about that later as well. But after telophase 1, we've got 2 haploid cells that have 2 sister chromatids for each of those chromosomes. In prophase 2, we start the process over again. We make a new spindle. In metaphase 2, now the sister chromatids are all going to line up at the metaphase plate. Again, if this was mitosis in the same organism, there would be 6 of these pairs of sister chromatids lined up, not 3. At anaphase 2, they're going to get pulled apart. And then in telophase 2, we're going to have 2 daughter cells for each of these parent haploid cells. So a total of 4. But there's no sister chromatids anymore. There's no homologous chromosomes anymore. Haploid daughter cells with 1 copy of each chromosome, unduplicated chromosomes. Okay, so as if that wasn't enough for giving us genetic diversity, these 3 things that we're going to talk about now really make the shuffling of the deck all the more certain to provide new combinations of genes in the next generation. So what happens during meiosis is, remember, part of the point of meiosis is to produce cells that are genetically different from the parent. And not just in the number, but in the what forms of genes appear in the daughter cells. In mitosis, the opposite is true. You want the products of mitosis to have exactly the same genetic information, exactly the same number of chromosomes. But these 3 mechanisms I'm going to talk about now are going to increase genetic variation. Independent assortment, crossing over, and genetic recombination, also known as random fertilization. Okay, so what is independent assortment? What this means is that when those homologous pairs of chromosomes line up in meiosis, which side of the metaphase plate they line up on is not consistent. So it's not like all of mom's chromosomes line up on one side and all of dad's chromosomes line up on the other side. It's a random assortment of things. So you might have, for your 46 chromosomes, on one side you might have 22 sets of mom's chromosomes and 1 set of dad's, or it might be 23 and 0, or it might be 13 and 10. But each of those homologous pairs is going to line up at the metaphase plate and separate independently from each other. So how many possible combinations are there? It's 2 to the power of n, wherever n is the haploid number. So for us, with 23 haploid number, there are more than 8 million possible combinations of chromosomes. Do I have a picture of this? I hope I have a picture of this. Yes, good. Okay, so at metaphase 1, in a simple organism that only has a diploid number of 4, haploid number of 2, here they are lining up at metaphase 1 at the metaphase plate. Now it's possible that at the metaphase plate, both of dad's chromosomes line up on one side and both of mom's chromosomes and line up on opposite them. But it's also equally likely that dad's going to be on the left for the first chromosome and mom's going to be on the right for the second one. And mom is going to be on the left for the second chromosome and dad's going to be on the right. What did I say? That it's going to be like this and not like this, simply put. So when those cells divide in metaphase 2, we're going to have different combinations of daughter cells depending on how they lined up at metaphase. So you can see we end up with either a pair of all blue cells and a pair of all red cells, or complementary arrangements of blue and red. And that's just for an organism that has 2 chromosomes. When we have 23, that means that if we wanted to show all the possibilities of how these things line up, then there would be 8 million different combinations for our pairs of chromosomes that are lining up from mom and dad. Okay, so another way that we get genetic diversity is from a process called crossing over. Crossing over produces chromosomes that didn't even exist in grandma or grandpa, because you can take chromosomes and arms of the chromosomes, so the lobes of the chromosomes, can physically attach and detach from each other. I'll show you a picture of that. This happens in prophase 1, when the homologous chromosomes pair up. Portions of the sister chromatids can trade places. And that means that a chromosome that has some of grandma's and some of grandpa's DNA in it, they may exchange arms with each other, and your mom might give you a chromosome that has a little bit of grandma, a little bit of grandpa in it. So let's see what that looks like. So this is all happening in prophase 1 of meiosis. The homologous chromosomes are lined up, and they'll start sort of forming these X-shaped structures called chiasmata, or just one is a chiasma. And you can see what's happened here is this blue chromosome now has a little bit of red at the ends of one of those sister chromatids. And the complementary version has happened over here, where you've got a little bit of blue on this red chromosome. Oh my goodness. And now look what happens at the end is we've got not just 2 different types of cells, but all 4 of our cells now have different chromosomes, are genetically distinct from each other. It's not just we have 2 blue and 2 red. We've got 1 blue, 1 red, 1 red and blue, and 1 blue and red. So yeah, this kind of makes the number of, imagine that, in our deck of cards analogy, that some of the suits were able to exchange dots. So you could have instead of having the jack of clubs, you could have the jack of chlorades or something like that, because it was a little bit club and a little bit spade. So crossing over also increases genetic diversity. Finally, random fertilization or genetic recombination means that while all of those processes go into making the gametes, the sperm and the egg, which sperm meets with which egg is also a random process. So the fusion of 2 gametes Remember, each of them is going to have about 8.4 million possible combinations from independent assortment alone, not even taking into account the crossing over, means that 8.4 million on mom's side, 8.4 million possible on dad's side, means that a zygote, even without crossing over, could have 70 trillion different combinations of all of the genes from the parents. That's a lot. Considering that world population at present is 7.5 billion. That is 7.5 with 9 zeros after it, or 8 zeros between 7 and the 5. And 70 trillion is 7.0 times 10 to the 13th. So there's enough genetic diversity just from independent assortment and random fertilization, not even including the crossing over, to account for 10,000 times the Earth's current population. So the chances of a genetic copy of you just popping up somewhere are pretty remote. Pretty remote. You are literally 1 in 7.5 billion. It's a lot better than being just 1 in a million. We are all genetically unique, and this is why. And there's something that's very special about that, don't you think? I mean, that you are a genetically unique individual, as am I. It's great, but it has its drawbacks. And I'm just, every time I think about it, I kind of marvel at it and this process that makes us all unique. So that is all I have to tell you about Chapter 5. And we're going to keep talking about DNA and genetics for a little while after this.